Bismillahirrahmanirrahim. We start the video 5 on the chapter on genetic technology. And this is A2 level biology 9700. We're going to discuss gene therapy, cystic fibrosis, and somatic and germ cell gene therapy. Now, what happens in gene therapy? Number one, we've identified the gene for the genetic disorder. Like in sickle cell anemia, there is the HBS allele which is defective. So a person with HBS is going to be suffering from sickle cell anemia. Now if we can identify that, okay, this is the problem in sickle cell anemia, now what we do is we want to find out the normal allele. Now you know the normal allele for it is HBA, which happens in normal people. And most of us would be HBA. So we find out the normal allele and then we use a vector like a virus to transfer the normal allele into the body cells and into the body cells where hemoglobin is being made. So what will happen is that instead of the defective hemoglobin being made now, the normal protein, the normal hemoglobin molecule will be made, and of course all the problem will be solved, because the person who has sickle cell anemia produces a hemoglobin molecule which has one amino acid which is wrong. So if we can introduce the normal allele for it, then the normal hemoglobin will be produced without the defective amino acid. So the person is not going to suffer from anemia. So in a way, we have found a cure. But then the trouble is that this probably will have to be given frequently. It can't be just one-off dose and then he's going to be okay for the rest of his life, but it'll have to be given repeatedly. Now, if you look at what I'm trying to say is we use a vector. The vector can be a virus. It can be a liposome. Liposome is what? It's a circular bit of like a little ball made up of a phospholipid bilayer. So the phospholipid bilayer will then fuse with the cell membrane of the human cell or the animal cell, wherever we want to use gene therapy for, but we're usually using it only for treating human diseases at the moment. So a liposome is a little sphere of DNA, like you would have a box made of cardboard. Here it's a phospholipid bilayer, and then in this you put the correct normal allele, which is the DNA sequence. And this of course will be carried. So the liposome will go and fuse. So the liposome will go and fuse with the body cell. And this correct allele or the DNA strand will be delivered into the body cell. So this is the body cell, this is the human body cell which needs this. And when this correct allele, which is all, please remember, this is the DNA. So this is the DNA strand, this is the DNA which enters the body cell. And then of course what is going to happen is that it's going to get attached to the DNA. And when it gets attached, now I've shown you 2 or 3 diagrams here. One, how a virus can be used. Second, how a liposome can be used. And basically what are we doing is we're delivering the correct DNA. So the correct DNA will come and become part of the host DNA, which is, now I've shown it to here. And then of course what is going to happen is that on the RER, we have the ribosomes. So the correct protein will be made here now. Now that is going to be very helpful, because whether that protein is a hemoglobin molecule, or whether that protein is an enzyme, or whether that protein is a channel protein, well, the correct protein will be made and the person will be cured and he'll be treated. So we use the vectors, we use our viruses, liposome, and at times just naked DNA is also used. And how it is taken to the cells, of course, that's another thing that we have to solve. Sometimes we give it in this form of inhalers, because if the disease is of the lungs, then the inhaler will correct the inhaler. When you inhale that, the correct allele will reach the lung cells and the correct protein will be made. So this is one of the ways in which we are going to start doing gene therapy. At the moment we have not been very successful, but I'm sure in the years to come all the problems will be solved, and this could be a very, very good way of treating the genetic disorders. We have just discussed gene therapy. And in order to talk about that a little more, I would like to discuss a disease called cystic fibrosis. Now, this is a disease which is found more in the European countries because we don't seem to have the allele for it here in the Asian countries, Pakistan, India, Bangladesh. Now, the first thing you've got to understand is I said it's an autosomal. Autosomal means that in a cell we have 23 pairs of chromosomes, but the 22 pairs are called autosomes. And the 23rd pair, which is an X or an XX or an XY, that's called the sex chromosome. So the 23rd pair, this is the 23rd pair. Now the 23rd pair is called a sex chromosome. If there's something on that, then we call it a sex-linked disorder. But I'm sorry, this is not a sex-linked disorder. So it's on the 22 chromosomes. Somewhere on the 22 chromosomes we have this. Now, of course, we also know it is on chromosome number 7. So if you take out the chromosomes and arrange them by height, we number them 1, 2, 3, 4 till 22. And we said it's an autosomal recessive. Autosomal recessive means it's on those 22 pairs of chromosomes which code for the body characters. Autosome is for the body chromosomes, and they do not decide the gender, but they code for all the other proteins which are necessary for the normal functioning of the human body. Now, if I say it's recessive, it means that would be homozygous recessive. You see, the other possibilities would be this. Now, this would be homozygous dominant. These people are not sufferers. This is heterozygous. These people are also not sufferers. But the poor people who are sufferers are the autosomal recessive ones who've got the two small a alleles. That's how we represent it, small a. Chromosome 7, and it is a gene that codes for a transporter protein. What does it mean? A transporter protein means that there is a cell membrane and we have a channel protein on it. Now, this channel protein is called CFTR. CFTR, cystic fibrosis transmembrane conductance regulator. CFTR stands for cystic fibrosis transmembrane conductance regulator. Now, if you try to understand that, what is it? We have a cell membrane, and in the cell membrane we have a channel protein, and this channel protein is very specifically for chloride ions. So what happens is, number one, it's an ATP powered pump, so it's an active transport pump. And what does it do? It pumps the chloride out of the cell, and these are usually the cells in the lungs. But when the chloride ions are pumped out, this lowers the water potential, so water follows, and the water comes and is lying on the outside of the cell. But then, if you remember, in the lungs there is something known as mucus. The mucus that is produced is viscous. But because this water moves out, the mucus becomes less viscous. So now what is going to happen? Now, what do we have? Who are the people who are the sufferers? The sufferers do not have a channel protein. There is no chloride ion channel protein. So normal cell, but when this chloride ion channel is made, it's defective. So this is not, it doesn't go, and because the cell machinery realizes that it's an incorrect protein, so it doesn't get embedded in the cell membrane. So there is no channel protein in the cell membrane. So what is not going to happen? No chloride ions are going to be pumped out. No chloride ions pumped out, no water follows. So the mucus is going to be pathetic. Well, how is it going to be pathetic? It's going to be highly viscous. Highly viscous means that it will not be able to be moved by the cilia. So if the cilia cannot move, it's going to collect there. And you know, mucus is a glycoprotein. Mucus is a glycoprotein. And glycoproteins are food for bacteria. So the mucus cannot be swept by the cilia. The mucus stays there in the bronchioles and bacteria multiply, and this person is going to be repeatedly suffering from lung infections. Plus, of course, also digestive issues, because the pancreatic duct gets blocked by this highly viscous mucus. So we have a lot of problems everywhere. Lung infections, food not being digested, because if the pancreatic duct is blocked, that means the enzymes cannot reach the duodenum, and the food that the person eats is not going to be digested. Now, if you look at cystic fibrosis, we just said it is a autosomal recessive disorder. So cystic fibrosis is most common among people of European ancestry. So that means they must be having the allele for it. We in Asia must be all having this allele. So we don't have this. We don't even have these people here. These people are not present in this part of the world. And the most cases result from a deletion of just 3 bases in the gene for a protein called cystic fibrosis transmembrane conductance regulator. As a result, the amino acid phenylalanine is missing from the protein. So there's one of these. You see, there are many different types of problems which result in the chloride ion channel being defective. You see, the chloride ion channel normally should be like this, say. Now the ones which are like this also are defective. The ones which are like this are also defective. The ones which are like this are also defective. So there are different alleles for it, which all result in the cystic fibrosis protein, CFTR protein being made, which is non-functional. So this channel protein is not going to go, and there's going to be no channel protein, or even if it is there, it's non-functional. So cystic fibrosis, homozygous recessive, autosomal on chromosome number 7. Another diagram to explain this is this protein, this channel protein is found in the surface of cell surface membranes of cells and tissues throughout the body, including the alveoli. So transport channel proteins allows chloride ions out of the cell and produced by the CFTR gene, and the location is chromosome 7, and it is 250,000 bases long. So you can understand what we are talking about. We are talking about this defect which is in the DNA. So this person who is born with this disorder would have the defect in the zygote when this person, the beginning of this human being, and then the zygote develops into an embryo and then a fetus and is born, and this person has this disease. Now, some of the symptoms of this child having this disease is a salty tasting skin, chronic respiratory problems, lung infections, poor growth, weight loss, and meconium ileus. So poor growth and weight loss is because the pancreatic enzymes cannot travel and digest the food. So what do we need to give these people? We need to give them enzymes with their food, so that those enzymes then digest the food which he or she eats. Respiratory problems, why? Because the mucus is not moved by the cilia, so the mucus stays in the bronchioles, and that results in bronchial infection, repeated lung infections. And it said chronic. Chronic means which has been there for a long time, like the opposite of chronic would be acute. But here we are saying chronic respiratory. So the poor child will be suffering from chronic problems for the rest of his life. Of course, they don't live long. Their average lifespan is about 35 years. Now, before all these genetic tests came into being, initially when we suspected somebody with cystic fibrosis, we would do a sweat chloride test. And the sweat test measures the level of chloride in the sweat using a small electric current. Pilocarpine, a chemical increases sweating. Mild electric current is given. The sweat is collected on a gauze for 30 minutes and then weighed in a weighing jar. So chloride more than this means it has cystic fibrosis. So this was called the sweat chloride test. Why? Because these sodium chloride, which is usually present in the sweat, is a normal level. Now this chloride ions cannot be absorbed by the cells in the sweat glands. So they have more chloride ions in the sweat duct. So more in the sweat. And this of course was a test that we would do, and these children would have, these babies born would have a very salty skin. So the sweat chloride test came into existence. Then overview again from the book that we used, prescribed by Cambridge. In a normal cell, the loss of chloride ions pulls water with it by osmosis. This keeps the surface moist and well lubricated. So here you can see the channel protein. You can see this is the outside of the cell. This is the inside of the cell. The chloride ions are pumped out of the cell, and then water is going to follow. And this of course would make the mucus less viscous. Now if this chloride ion channel is not working, like it's made something like this, well, it's non-functional, then it's not going to pump out the chloride ions. So the chloride ions, if are not pumped out, water does not follow, mucus highly viscous. Now let's look at gene therapy for cystic fibrosis. So what we do is we take a virus, and we take a virus which usually infects the lung cells, like the influenza virus, and we of course deactivate it so it doesn't cause the disease. And what we do is we attach the normal allele, the normal DNA sequence, which is for the CFTR protein. And then we hope that this virus is going to enter, and then this virus will go and get attached to the virus, will enter, the DNA will follow, and the correct allele DNA will go and get attached into the nucleus with the chromosome, and hopefully we are thinking that the normal channel protein will then be produced and chloride ions would be pumped out. But this has not been a very success story, because what we did is we gave it in the form of inhalers, but then the normal DNA, which we use the virus as the vector to take it into the human cells, somehow got attached to a part of the chromosome where it resulted in uncontrolled cell division, and those people developed lung cancers. So it's not been a very successful story. We might even throw the DNA into the human body in a liposome, or we might do it just in like a naked DNA. But then what we do not realize is that it doesn't go and just result in the formation of the protein, and then thus the protein being formed, but it sometimes gets attached, that DNA gets attached to some other part of the chromosome, and that results in uncontrolled cell division. Moreover, you need to take this daily, because you need to spray. Like if you take it in the form of an inhaler, well, you will have to take it very frequently, maybe every second day or every third day, because that DNA does not sort of become part of the host DNA as a permanent feature. It's going to be degraded and it's going to be lost. So you need to take this inhaler every third day or every fourth day. So this is one of the ways that we can do gene therapy for cystic fibrosis. Now, a genetic diagram explains to you how two people both have to be heterozygous. Both have to be heterozygous to have a child who is a sufferer. Now I give you a very simple example. Just for a minute I give you my own example, and I say, okay, well, my brother's son is a sufferer. So if my brother's son is a sufferer, that means my brother is this. He's heterozygous. Now, what can I be? Either I'm this or I'm this. Now, would I like to know this? Well, of course I can if I go for genetic screening. But then the question is, do I want to really know what I am? Because I have seen my brother, having this child who is a sufferer, and I see him going through the pain and the agony of bringing up this child. And now I know that if I do genetic screening, I can probably figure it out. What am I? Am I this or am I this? Now, if I'm this, I have no way to change my genome. So really, if That is what one of the ethics of genetic screening we talk about, is that you have no cure for it, no treatment for it, and getting to know that you have this in your genetics, is that any help or not? So these are the questions which I want you to think about whenever we are doing the ethics of genetic screening. Now coming to the topic of somatic and germ cell gene therapy. Somatic means body cells. So if we are sort of targeting the body cells and we are introducing some sort of a DNA which is carrying the correct allele, well then we're just targeting a few body cells, and we are talking of taking the allele and then of course resulting in the correct protein being produced, whether it's an enzyme or a channel protein. That depends on what the disorder we're talking about. But when we are talking of germ cell gene therapy, we are either going to alter the sperm, or we're going to alter the ovum, or we're going to alter the zygote. Now, what we need to understand is when we're altering the sperm or the ovum, finally what we're getting is we're getting some allele that we have introduced into the zygote. Now, when a human being develops from this zygote, first of all, it forms a ball of cells. Now, every cell of this person will have that allele. And then, of course, when that person grows up, and he would have then in his reproductive cells as well, if he's a man, then he'll have it in the sperms, and if he's a woman, will have it in the ovum. So, are we okay with that, or are we really not okay with this? Because we don't know what could be the effects. So doing germ cell gene therapy is a very controversial topic, and a lot of discussion goes into it and whether we should do it or we shouldn't do it. Discussing germ cell gene therapy, the example is, for example, in theory, a woman with cystic fibrosis could opt to try to conceive a baby using IVF. So the eggs or the oocyte would be harvested from her in the normal way. So we would from the ovaries extract the oocytes. Then the correct allele of the CFTR gene could be injected into an egg, and then this egg fertilized by a sperm to produce a zygote. Now at present this is illegal in humans. However, it has been successfully done in other animals, although it looks as though there is a high chance of offspring produced in this way having other unpredicted diseases. So this is still, germ cell gene therapy is still under discussions and whether we should do it in humans or not. Because when we're doing this germ cell therapy, all the cells of the child are produced from this genetically engineered zygote, and therefore will all carry the gene that has been inserted. When the child grows up and produces eggs or sperms, these gametes will also contain the allele, and therefore it will be passed on to their children. We say that the allele is in the germ line, being passed on from generation to generation. So this is all what we need to be discussing under these topics, and with that I end this video, and inshallah I will complete this chapter in the next video, in which we're going to be talking about genetic technology and agriculture. And thank you for now. In this video of the video 5 of 8.2 biology, we discuss gene therapy, cystic fibrosis, and somatic and germ cell gene therapy.